southwestern and northwestern screening. In previous technique, we discussed that how bacteriophage lambda GT11 or lambda ZAP vector can be used for the screening of cDNA libraries that will produce a expression product. So one of the modification of the previous method, it can be used for the detection of DNA specific binding proteins or RNA binding specific proteins that may be referred as southwestern or northwestern blotting. मेरा ख्याल है कि आपको याद होगा कि स्टार्ट में हमने डिस्कस किया था अबाउट द सदर्न ब्लॉटिंग। इसमें हम डीएनए को एग्रोस जेल पे रन करते हैं सो दैट द फ्रैगमेंट्स में बी सेपरेटेड, so that they may be purified finally, and the separated DNA fragments, they are shifted to nitrocellulose membrane, and after shifting to nitrocellulose membranes, they are immobilized, and then by using a specific probe, by using hybridization method, the target sequence, they are detected. And similarly, this technique, it can be used for RNA, and this technique, it is called as northern blotting. इसी तरह से वेस्टर्न ब्लोटिंग टेक्निक भी हमने डिस्कस की, जहां हम डायरेक्टली RNA तो यूज़ नहीं करते, रादर प्रोटीन फ्रैगमेंट्स को यूज़ करते हैं दैट हैव बीन सेपरेटेड बाय पॉलीएक्रिलामाइड जेल इलेक्ट्रोफोरेसिस। तो प्रोटीन फ्रैगमेंट्स को हम शिफ्ट करते हैं नाइट्रोसेलुलोज मेम्ब्रेन पे, एंड देन बाय यूजिंग स्पेसिफिक एंटीबॉडीज the proteins they are detected, and this technique we called as western blotting. So one of the modification of all these technique has been used to detect DNA or RNA binding proteins. For example, for southwestern blotting, what we can do that target DNA, it can be immobilized on the nitrocellulose membrane, and here the antibodies, they are not used, and then the expression proteins that may be DNA binding proteins, they are incubated together, and if they have target sequence affinity, then the proteins, they may bind with double stranded DNA on the membrane. So because of this reason, here we are combining the principles of both the southern and western blotting. We are calling this technique as southwestern blotting, and this technique, it is very valuable to screen the transcription factor from different cDNA from mammalian cells. So this is one of the advantage of southwestern blotting. Similarly, we can also use northwestern blotting, where we can immobilize the RNA molecule on nitrocellulose membrane, and then they are incubated with expressed protein products to find the homology between the RNA and protein sequences, that they are having affinity with each other. So in this way, southern western blotting or northern western blotting, they can be used to detect specific DNA molecules.